Well, hello everybody, and welcome to Ovarian Cancer Australia's new initiative, our information and support webinars. My name is Anne-Marie Ferguson, and I am one of the coordinators of the support programs. Before we begin, I would like to acknowledge the traditional owners of the lands that we're standing on, the Australian Indigenous people, and I pay my respects to their elders, past and present, and any that may join us today. I'd also like to thank the Brockhoff Foundation for providing Ovarian Cancer Australia with a funding opportunity to deliver this project. In fact, we have women from all over the country joining us today. Our webinars cover a range of topics that are actually specifically tailored for women with ovarian cancer and their support networks. And today's webinar is on clinical trials and research. This webinar is live and interactive. You can type questions you may have for the presenters in the chat box, which is located at the bottom left-hand side of your screen. And please be aware that our presenters won't be addressing any individual medical cases, but you can call the Ovarian Cancer Australia Information and Referral Line to speak to one of our specialist support teams after this webinar. If you're experiencing difficulty hearing the sound during the webinar, please dial the 1-800 number that's listed in the chat box. Now, I'd like to begin with welcoming our presenters for today, Dr Yoland Antill, medical oncologist, and Jackie Polonsky. Dr Antill is a fellow of the Royal Australasian College of Physicians. Yoland is the lead investigator for a number of local and international research studies and is involved in the development and conduct of clinical trials. Yoland has more than 10 years' experience in both medical oncology and in the diagnosis and management of hereditary cancer syndromes. Jackie is a woman living with ovarian cancer and is passionate about improving the lives of other women living with the disease. Jackie is on a clinical trial and feels very strongly about promoting awareness about ovarian cancer and how to live well with the disease. I just want to say a special thanks to Jackie for joining us at such short notice. I'd like now to pass you over to Dr. Yoland Antill to begin the presentation. Thanks, Yoland.
So welcome everybody today. I'm assuming that everyone can see my first slide there. So I have been asked to talk today about clinical trials and research in the setting of ovarian cancer. I am a medical oncologist and I am also a cancer geneticist, which means that I look at people or see people and their families in relation to gene testing to see whether they have an inherited risk for cancer in their families or as a cause of the cancer they might have. I'm also a clinical researcher, so I, in my hospital sites, look after clinical trials, both in the area of breast and other gynecological cancers as well as breast cancer. I'm also a cancer researcher, and hopefully by the end of my talk you'll understand a little bit about the differences between what a clinical trial is and what research is and where the overlap might be. So I guess the very first thing is where I wanted to start with is really trying to talk about the difference between research and how it differs from a clinical trial. When we have a medication or a drug that's available in the clinic to treat our patients and their cancers, It's a very long road to having this drug available for us to use. It starts down here at the design level. Someone comes up with a thought, and then that thought process is taken into the laboratory where it's tried in terms of understanding whether it might be applicable to the cancer in question. It then moves into a workable format. Can we actually make this potential thought into a drug that can be given? And can we test that in terms of cancer cell lines? And we'll talk a little bit more about all these steps in the coming few slides. Does it work in terms of treating cancers within animal models? And we'll talk about that before it even moves into a clinical setting. And our phase 1 clinical trial setting is the very first stages where a medication might be seen in the use for the treatments of cancers within humans. And if we see a positive signal, it's moved into what's called a phase 2, so it's a smaller number of people, before a larger multi-center clinical trial. And if all this is successful, then a pharmacological company will take this to government bodies, usually first of all the FDA in the US, to apply for an application for use in the clinical setting. And then once they get that go-ahead, that's when the marketing takes place. This is a very, very long road, and we'll go through some of the processes through this today. So taking a step back, cancer research starts with an idea or theory, and it moves quite quickly, sometimes, into the laboratory. In terms of cancer, the very first step is, in fact, to understand what is a cancer, what stimulates cancer cells to grow in the first place, and what stops the body's normal regulation systems from actually recognizing that these cancer cells are abnormal, and why can't they deal with them? Much like an infection, when the body recognizes that there is some bacteria there, it stimulates the immune system to, in fact, knock that infection on its head. So I guess I'm passionate about food, so I like to bring my analogies back to the kitchen because I think it's something that we're all quite familiar with. So what is cancer? Let's start in the kitchen. If we think about the process of washing up is a very ordered process. We have a pile of dirty dishes on one side. We have our soapy sink full of warm soapy water here, and we take a dirty plate, we clean it, we rinse it, and then we dry it. And it's a very ordered process. So I'm not sure how many of you have children or grandchildren or who have ever seen the film Sword in the Stone, but this scene in the Sword in the Stone really reminds me of what goes wrong in cancer. So Wart is the youngest and the main character in the book, who goes on to become King Arthur, who's asked to wash up all these dishes. And then Merlin comes along and casts a spell to automate the process, or he interferes with that solitary movement of dirty dish, water, drying, and back onto the shelf. And what happens is that process of automation, suddenly something goes wrong, and everything becomes very, very chaotic, and everything, in fact, falls apart. So if we take that concept of what was an ordered process back into our science lab, and we think about a normal, healthy cell that would normally go through a process of division from one cell into 2 new daughter cells, and so on, if in fact we have an injured or an abnormal cell, normally the body would be able to recognize that, and it would make that cell die so that it can't go on to produce abnormal cells. So what happens in cancer is that something goes wrong with this recognition of this abnormal or injured cell. And in fact, this abnormal cell is allowed to produce and goes on to produce more and more abnormal cells. And when that happens, in the generation of cells to come, these cells become even more abnormal and even more automatic in their process of growth and no longer able to respond to the normal stop measures that would be there to prevent cells from being able to grow and divide and to make new cells. All this ordered process is driven largely by genetic regulations. So most of us have heard about genes by now. So what is a gene? A gene is something like a protein or a substance that has control or regulatory control. How does a gene function? A gene, in fact, is a very long string of letters or a code, much like when you're making a cake. If you have a whole lot of listed ingredients and you use the ingredients in the right format and the right order in your recipe, and you read it from beginning to end, you come out with the cake or whatever at the end of it. But if you don't read your gene properly, if the ingredients are in the wrong way, or if you have a substitution, for example, plain flour instead of self-raising flour. So in terms of genetic code, if you substitute the wrong letter in for a letter that was supposed to be there, what happens is a massive flunk. The cake doesn't work. So if we go back to the gene and we think about here is our cell, here is the central powerhouse of the cell called the nucleus, and this is where all our genetic material or gene material is found. And if this gene is read from beginning to end in a normal fashion, it produces a normal protein that is allowed to function either as a stimulant of cell growth or an inhibitor to stop those cells from growing or expressing. And genes are responsible for pretty much everything that we do, from eye colour, from hair colour, although the fashion industry and cosmetic industry is doing a lot to, in fact, inhibit the normal genetic expression of hair colour these days. It tells us whether we're going to be tall or short, whether we're going to be a runner or a swimmer, whether we're a scientist or someone who is more artistic in their measure, even in terms of personality, whether you're someone who is quiet and happy to be by themselves, or whether you are just in your comfort zone surrounded by noise and other people. But genes and errors in genes can also lead to disorders or disease, including cancer. So let's bring this all back. Here is our normal cell, and here is it normally dividing, and here is our injured cell that would normally have this protein to act that's been produced by the gene to act here to recognize this is abnormal, and it will call in the normal regulatory processes in the body to make that cell die. But if the gene isn't working, or genes isn't working properly, then this protein or stop gap isn't here, and it allows these tumour cells to go on and regulate and grow in an unregulated fashion. The other really important part to understanding about tumours and cells and how they talk to each other is this concept about cell signalling. So here is our beautiful cell, and you can see in the middle of this cell is the nucleus. This is where that genetic material is that tells the cell how to behave and what to do. You can see here that this is a 3D picture of a cell. It's a cartoon, and in the middle is the nucleus. And then surrounding the nucleus is all these highways and pathways that either provide power to the cell or that it allows these signals to be sent from the outside of the cell or the inside of the cell to the outside, and so on and so forth. So this is a little pictorial blow-up of what happens at this cell surface. You can see that there's a little signal or a protein trying to tell this cell what to do. And what it has to do is land on what we call a receptor that activates a signal on the inside of the cell and sends that message to the nucleus, the powerhouse of the cell, that then tells the cell what to do. So in terms of developing drugs, we can take advantage of this, because what happens in cell regulation for cancers is that this cell signal pathway that would normally respond to saying either I am an abnormal cell, and the bodies would come and say, well, we don't want you to grow here anymore, so it cuts off that signal pathway, or we are a tumour cell and we're going to overcome the normal body's regulation, and it upregulates, it uses the body's own hormones or proteins to in fact cause its own cells to replicate or grow and divide. So if we block these normal signal pathways that can activate a cancer cell to grow, or stops the normal body systems from inhibiting, then we have a chance of targeting the very thing that might make a cancer cell grow or stop a cancer cell from being recognized by the normal body's immune system. And it's these cell signal pathways that have become the focus of research now in the setting of treating cancers. But these pathways are really complicated, and it is not just one cell signal pathway that causes any one cancer to grow or divide. There are many, many pathways and many back roads into the same pathway that can either be blocked or overcome or upregulated. This is a pictorial pathway of literally one signalling pathway. And cancer cells will often have even up to 10 different signalling pathways that it can use to keep its mechanism for cell growth to keep itself from dying. So where are we in terms of this road to discovery? We are here. So understanding all that background to how a cancer cell grows and how it actually sends these signals out to keep growing is all in this design pathway. It's this background research that leads us into the setting or the pathway to clinical trial, clinically trialing medications that might be used to treat a cancer. If we now just spend a little bit of time thinking about the phases of clinical trial. So what I've talked about is, so far now, is this pre-clinical setting. This is this understanding, trying to understand what makes a cancer cell grow, what stops a cancer cell from growing, what agents do we have to use here, how can we design a medication to use specifically in one of those cell signaling pathways, and can then we use that in the setting of cancer treatments. So what happens after the idea is that we have what's called cancer cell lines, and we can reproduce these cancer cells in what we call cell culture. And these are cells that we can grow in the laboratories, and we can test different medications on those cells. The problem with cell culture and being able to make these seemingly endless supply of cancer cells to test our cancer treatments in theory is that they often lose the normal regulatory signalling and pathways that a cancer within a human has. So they don't quite behave the same way as it does either within an animal or especially within a human. So once you have something that is looking like it could be a goer, could be a positive treatment option, we do use usually mice models, and tumours grow in the mice, and we test the cancer agents in those animal models before they're ever used in humans. The regulations around the use of animals in the development of medications for the treatment of cancer is very, very carefully regulated by ethics bodies. Many people are very aware that this is a pretty sensitive issue. So once we've gone beyond this preclinical phase, then we move into what's called a phase 1. So these are in human or in people studies who have cancers. They are effectively about finding the right dose. So it's that balance between finding the dose that is effective against fighting the cancer versus not causing too much or if any in the way of side effects or toxicities. So this is our dose finding signals. Phase 1 studies are often about collecting groups of patients who might not have any treatment options remaining to them amongst the generally available treatments, but who remain well and who want to remain on some sort of possible treatment that may benefit them. But there may not be benefit from a phase 1 study. A phase 2 study is where we see a positive signal in a particular cancer. We have a larger number of people, around 100 to 150, usually in a small number of centres, to test the theory of this individual therapeutic option. And then again, if this is successful without too much in the way of side effect, it moves into a much larger study between 300 and 3,000 people to evaluate the safety. So is that signal still valid, and are we sure that it's going to be safe to give to people? Once this, again, is a successful study, the regulation application applies here, and then once it has been approved for active use in humans in certain cancers, then it moves into that post-regulatory approval or phase 4 setting. This all takes a very long time. So if we think that we start out potentially with around 5,000 to 10,000 different compounds, we move 250 into that preclinical phase, and of these 250, 5 of them might move into early phase treatments, and even a smaller number will then move into these larger phase studies. Then this was what it takes to get one single drug from idea to market. And the cost of this is approximately $2.6 billion over 10 years. We have to get better at finding ways to get successful therapeutic options into the clinical pathway. We have to be smarter about how we are going through that research phase into clinical trial. Some things to think about. All these clinical trials are generally done with the most common sort of cancers. So in terms of ovarian cancer, we see many more trials treating high-grade serous or high-grade endometrioid or high-grade clear cell. But if you have a high-grade mucinous cancer, there are almost no clinical trials that you will be eligible for. So what if you have this rare type of cancer? How do you find a clinical trial that might be appropriate for you? So most clinical trials are aimed at the more common cancers for a couple of very obvious reasons. One is that largely these research and the clinical trials themselves are funded by pharmaceutical companies. So they want to treat the more common of cancers, and so in order to recover the costs of research and development, they will stick with cancers where a larger number of patients are able to be gathered more quickly to effectively find the answer of whether this is a successful treatment or not. The other thing, of course, is that if you have a rare cancer, you might be one person in one state in 1 year. And in order to get sufficient numbers of people around the world to get the answers for these clinical trials, it might in fact take hundreds of different institutions to gather the right number of people over many, many years. And that's just not a cost-effective way of being able to trial these drugs. Some of the other things to think about is that my doctor told me that my cancer might respond to a type of therapy, but these trials that are currently being run are in all sorts of different cancers. For example, if you have a mutation in one particular signalling pathway, then that application might be used for breast cancer or stomach cancer, but isn't for ovarian cancer. So one of the new ways of thinking about clinical trials is in fact doing what we call a basket trial. So we are testing this theory of one signalling pathway, for example, the HER2 pathway, which we know might be amplified in a whole range of cancers. And so what they do is they're testing this one agent in a number of cancers, which is a more efficient way of testing the agent for cancers, all cancers that have this particular pathway. So one of the other options that we're just working with at the moment is this concept of tag-on. So this is a very old trial and it was done many years ago, and I keep using HER2 as an example, but it's just an example I pulled out of the air. And the HERA trial was looking at the use of a medication called Herceptin for breast cancers that were HER2 amplified. But we know that a number of other tumours will have HER2 amplification or mutations, and that they may well be susceptible to this anti-HER2 therapy. So if you set up a big breast cancer trial, can you in fact put in some tag-on positions in the trial that would allow these rare cancers to fit into this tag-on position, to receive the medication in a regulated fashion that monitors the outcome, doesn't affect the overall results of the breast cancer study, but still enables researchers to be able to look at this concept of rare cancers who might be amenable to these types of treatments, and then gather the information from around the world as tag-on for this specific agent. So who runs clinical trials? There's no doubt that the pharmaceutical industry are the major sponsors for these clinical trials. Not so much the preclinical research, but the actual movement from preclinical phase into the clinical phase. And these are just a number of the pharmaceutical companies. It's certainly not all of them, and they're not favoured. I just pulled them out of the hat. Researchers can apply to these pharmaceutical companies with an idea. So they are what's called an investigator-driven trial, where a pharmaceutical company might provide the funds to run the entire trial, or in fact they might be happy to provide the drug, but the remainder of the funding for the trial has to come from other areas. ANZGOG in Australia is the Australian-New Zealand Gynecology Oncology Group, and works in collaboration with the National Health and Medical Research Council Trial Centre here in Australia. They are the brains trust of gynecological cancers here in Australia and New Zealand. And what they can do is to help coordinate and develop clinical trial or other research project protocol development. They are also a really great link into other international trials groups to be able to get clinical trials from that preclinical or that idea setting into the actual clinical trial process. So an investigator might go to a company and say, look, I'm interested in doing a clinical trial in X, Y, and Z. They can't generally do that by themselves, so what they might do here in Australia is go to ANZCOC and say, I think I've got a good idea for a trial. And that allows a group of experts to come together to help develop that idea into a clinical trial synopsis or protocol, which is then taken to a pharmaceutical company for either full funding or perhaps to please give us the medication in question and we will come up with the other funds. And then if it gets bigger than that, leading into those bigger international phase 3 trials by linking in with other gynecological oncology groups around the world. So what about other research? So I've talked largely about clinical trials with the use of therapeutic options available for the treatment of cancers. But there are many other parts of research that goes into looking after a patient who might have ovarian cancer. Epidemiological studies. Who is at risk of cancer? How did it happen? What are the risk factors? Are there hereditary factors? How can we make the surgery associated with ovarian cancer better? And there are surgical trials that go on all the time. How do we look after patients preoperatively? How do we look after them after their surgery? Is there any imaging technique that we can improve on? We know that ovarian cancer is a notoriously difficult cancer to see using standard current imaging techniques such as the CT scanner or ultrasound. We also know that screening techniques are really difficult to use, with imaging so far, even in combination with blood tests, not successful in being able to pick up the very earliest stages of ovarian cancer. How do we support someone? What are the supports that are needed by someone who has a diagnosis of ovarian cancer? Is exercise? This seems to be the real buzzword in cancer at the moment. So can exercise be something that's helpful for someone who's got ovarian cancer? Can exercise be used to improve outcomes after a big surgical procedure on the abdomen? Is meditation good? Nutrition? What's the impact of others? I've had both my ovaries removed, and this is really making me feel never like I was ever a woman to start with, or I'm feeling like I'm really different now, to feel different towards my partner in terms of libido. All these other things. Research goes on in all these areas to try and improve, help, and understand both the road that a woman might experience while they're being diagnosed and treated and looked after in the stages of their ovarian cancer. But how does all this research get funded if it's outside a clinical trial? And this is where you guys come in. Fundraising is the only way we get to go forward in terms of our treatment options, how we get better. And we know that ovarian cancer fundraisers are pretty good fundraisers. So for that, we are all very, very grateful. So I'm going to stop there. And I don't know if this is where I take questions, or are we going on to hear from Jackie now?
Hello, Yoland. Thank you. That was fantastic. I think what I'd like to do is just pose to you a couple of questions that's come through before we pass it over to Jackie.
Yeah.
Our first question, Debbie asks, when having a drug, how can a patient give feedback on side effects they are experiencing, but they're not listed as potential side effects?
So that's a really important question. If you're on a clinical trial, I'm sorry about my iPad, it'll stop ringing in a minute. If you're on a clinical trial, any side effect, whether it's expected or unexpected, is something that you would need to discuss with the person who is looking after you on that clinical trial, and that should be noted down and that side effect should be fed back to the research team. If you're experiencing a symptom or a side effect that might be related to the medication, but it's in standard use and it isn't something that you or your researcher were expecting, then we have the opportunity to go back to a pharmaceutical company and say, my patient has presented with this and X, Y, and Z, and I think it's a possibility that this may be related to the medication. That's called post-marketing analysis and safety. And that goes on all the time with medications. And there are pharmaceutical specialists that are employed specifically to look at the ongoing safety and monitoring of the medications that are used in the clinical setting. The most notorious example of that was thalidomide in the 60s. It was a drug that went through clinical trial phase very quickly, albeit we know now that there were some gross errors that were incorporated and allowed to pass through unaudited, and meant that there were some unexpected side effects seen, that it wasn't until that the obstetricians or pediatricians went back and said, look, there's a common theme here, that these babies that are being born with significant effect to their limbs, all their mothers were receiving a medication for morning sickness called thalidomide. And that's where these regulation processes keep going, even after a medication is allowed for clinical use.
Thank you very much for that, Yoland. Another question, from Haley. Thanks for the information, Dr. Antill. Can you give us an example of any new important trials which might be on the horizon in Australia?
Oh, where do I start? So I'll pick a rare cancer to start with. So the concept of mucinous or high-grade mucinous ovarian cancer is something that I've been involved in this preclinical work. So we're at that preclinical stage, and we've discovered that a number of these cancers have this HER2 pathway, and I guess this is why it came to mind when I was writing this talk. But it's a rare cancer. We might only see 2 or 3 in Australia in any given year. So we were trying to think of this process of how we might move into the clinical trial setting, and at the moment we are talking about this tag-on concept to some of the other trials that are using anti-HER2 agents. We also know that inhibiting a pathway called PARP, or PARP inhibition, is potentially particularly important for patients whose ovarian cancers have occurred in the setting of an inherited mistake in the 2 genes BRCA1 and BRCA2. But it may be that there are another cohort of patients or a group of patients whose ovarian cancers might respond to the use of those treatments. And they are, while they haven't inherited one of those gene errors, their tumours might actually have that acquired mistake in their genetic code in the cancer cells or the tumour cells themselves. So if your tumour has that mistake, could that be a treatment that is in fact effective? We know that there are a number of trials that have in fact just closed looking at hormonal agents or hormonal blocking agents, particularly in the treatment of low-grade ovarian cancers. So there are a number of different both specific targeted treatments for different types of ovarian cancer, hereditary or non-hereditary types of cancer. But I guess the newest kid on the block, and I can see that there is a question down the list here about these immune-stimulating therapies, and they are, I guess, the real poster child of cancer therapies at the moment. I didn't talk about them specifically in my talk, but one of the ways, and again I'm going back to films because film is another one of my loves, and if you know the film or the series of Harry Potter, Harry has an invisibility cloak and he wraps it around himself, and while he can see and go on and act normally in the world, others can't see him or interact with him. And that's kind of like what a cancer cell does to evade or avoid the immune system's own regulatory systems. It's the reason why that immune cell can't say, hey, you aren't a normal cell, go and die. And what the immune-stimulating therapies do is take away the invisibility cloak to allow the immune system go, whoa, you should not be here, and allows the immune system then to attack the cancer cells. And there are a number of clinical trials that are open in phase 1, but will open in phase 2 or 3 in the coming months and years for the treatment, particularly of high-grade ovarian cancers.
Thanks, Yoland. Do you think that answers Julie's question, or shall I pose that question? Is there further you could say? Julie's question for the rest of the participants, she asks, are there any trials into the effectiveness of low-dose naltrexone on boosting immunity?
No, that's a different question. Not that I'm aware of, Julie.
Thank you. Thanks, Yoland. I'd like now to invite Jackie Polonsky to share her story, her experience with us. Jackie, over to you.